Okay, welcome back friends. Welcome back in another video tutorial from microbiology with Chagnik. So in that video tutorial, I am going to discuss about regulation of gene expression, that is the Operon model. Okay, so let's start our discussion. So okay, so first of all, before going to discuss about our regulation of gene expression, I am going to discuss about basic regulation of gene expression difference between eukaryotic cell and prokaryotic cell. Okay, remember the regulation and gene expression means simply the transcription, okay, of DNA into RNA. Okay, now this transcription is regulated by promoter, operator and terminator region. Okay, so remember one thing, that in case of eukaryotic cell there will be one promoter, one operator and one terminator or terminator region for a single gene. Okay, and in case of prokaryote, for many genes or multiple number of genes, there will be one promoter, operator and terminator region. Why this so? Because remember one thing, in case of eukaryotic cell means they are relatively larger than the prokaryote, right? So prokaryote have to complete their metabolism in a very confined area, right? So that is the reason they incorporate multiple genes, or they also sometimes incorporate single gene with a single promoter, operator and terminator region. Okay, but remember one thing, that it does not mean that they will incorporate two gene with one promoter and an operator, in which one will go for one metabolism and another one will go for some another metabolism. Okay, that will not happen. For example, in case of lac operon, okay, we have three genes. In case of lac operon, I am going to discuss with you all about the three genes. So these three genes are all regulated by one operon. Okay, but remember one thing, these three genes are all involved in lactose metabolism. Okay, there is nothing something like that number one gene who is regulated by lactose, lac promoter, is involved in lactose metabolism, and the second one is involved in tryptophan metabolism. No, you cannot find something like that. So okay, so that you need to remember that in case of eukaryote, a single promoter for a single eukaryotic gene, single operator for a single eukaryotic gene, single transcription terminator for a single eukaryotic gene. But in case of prokaryote, the case is different. Okay, so now let's move to the next slide. So this is the operon. Operon means what? Operon means the expression of gene is regulated. Okay, so the regulation of expression of gene with the help of that promoter, operator and transcription terminator is known as operon concept. Okay, so remember one thing before going to enter in deep of the operon, that gene can be divided into two parts. One is constitutive gene, another one is repressor gene. Okay, or you can say regulatory gene, not repressor, but more specifically it should say regulatory gene. Okay, so constitutive gene means, as its name suggests, these genes are activated all the time inside the cell. Okay, but in case of regulatory gene, the picture is different. In case of regulatory gene, remember it very clearly, we have operon system. Okay, that means MCQ may come for what kind of gene you can find operon system. So do we require any kind of expression, a regulation of expression in case of constitutive gene, because they are activated all the time inside the cell? Do we require? No. So but in case of regulatory gene, yes, we have. So answer will be regulatory gene in that case. Okay, so Jacob and Monod are the two scientists that first prepared the concept of operon. Okay, so another important concept that I want to give you here, that is the classification of operon. Okay, so operon, sorry, not coperon, it is operon. So operon can be divided into two operon. One is catabolic operon, another one is anabolic operon. Okay, remember one thing, catabolic operon means it metabolizes something. Okay, that means with the help of this operon there will be conversion of some complex product into simple and usable product. Okay, suppose in case of lactose operon, remember one thing, that lactose operon is the example of catabolic operon. Here lactose operon helps in the conversion of lactose into glucose and galactose. Okay, that can be readily used up by the E. coli. Okay, but in case of anabolic operon the picture is different. An example of anabolic operon is tryptophan operon. Okay, so remember one thing, anabolic operon means here there will be some synthesis of new product. Okay, so with the help of tryptophan operon, remember that we are synthesizing tryptophan. Simple concept. Okay, so now you tell me that suppose there is a present of catabolic operon containing cell. Okay, and now whether you have to add the molecule in the medium whose conversion into smaller or simpler substrate, simpler usable substrate you are requiring. Yes, you have to add. If you don't add this, then how can those lac operon convert them into smaller glucose and galactose, right? So in case of catabolic operon, the presence of molecule in the medium is necessary for switch on of the operon. Okay, but in case of anabolic operon, remember one thing, anabolic operon is the backup plan for any organism. Okay, suppose you used to be very rich, suppose you belongs from a very rich family. Okay, but somehow unfortunately your family condition become very poor. Okay, so in that time you have the responsibility to take all the responsibility to earn some money for your family, right? So anabolic operon is the that children in the house of, or that children in a rich family that usually takes the responsibility of synthesizing something when the natural or actual source is somehow not available. Okay, suppose in case of bacteria, whenever it is growing in a system that have tryptophan, so it will not start its tryptophan operon mechanism. Okay, but when the media lacks tryptophan, okay, so it will start the tryptophan synthesis. Okay, with the help of tryptophan operon. So that is the idea. In case of catabolic operon, we can switch on the catabolic operon just by adding the molecules in the medium, right? But in case of anabolic operon, the thing is exactly different, or exactly the reverse. In case of anabolic operon, we cannot add any kind of molecules. Okay, otherwise the anabolic operon will not start. Now let's move to the next slide. So now we are going to discuss about the general structure of operon that we need to remember. So there are two parts, regulatory part and structural part. Okay, remember one thing, in regulatory part it lacks operator, but in case of structural part it have operator. Okay, and operator is generally just beside the promoter. Okay, first there will be promoter, then operator, then our gene whose transcription is regulated by RNA polymerase present in the promoter, and the transcription terminator region will be at the end. That is the normal order. But in case of regulatory part, the operator is not present. Okay, And another thing that you need to remember, that in case of regulatory gene, the promoter is weak. Okay. So now let's focus. Promoter is weak, so the transcription of those genes will be in very low concentration. Okay, with the help of that promoter. Okay, so this low concentration regulatory product will play a important role in regulating the structural gene. Okay, so there are two part in case of general structure of operon. One is regulatory part, another one is structural part. In case of regulatory part, operon operator will not be present. Okay, and promoter is weak promoter, and this weak promoter will generate regulatory product which is in low concentration. This regulatory product will going to regulate the structural genes that are present in the structural part. Okay, so that is the normal concept of the operon that you need to remember. Okay, so I am wrapping up this video here. In the next video we are going to discuss about the lac operon.